Hello, everybody. This is Giorgio Pea. I'm a product manager at Thermo Scientific. I cover AmpliSeq Designer, AmpliSeq HD, and AmpliSeq White Glove service. That's what I'm going to talk to you about, specifically as related to NGS solution for infectious disease research. As we all well know, unfortunately, pathogens pose a challenge. We have been through tough times lately, and they put us a challenge, and ultimately, we cannot risk to stay behind those challenges and in understanding what their, the pathogen dynamics is. And, just here to highlight and, recent or less recent example or representing pathogens that are coming up, not every day, but very often as of late. But, well, the good news is that genetics and NGS and advancement into research bring us with the capability to provide us hints and information about those ahead of time, so that could give us that little advantage over these pathogens and infectious disease agents that can make a huge difference, although small. And these, the application into next generation sequencing and exploiting next generation sequencing in infectious disease research give a widespread range of application going, as stated here, from epidemiology, where the main question there is, how does a pathogen evolve and spread in time or in regions? So we can track transmissions of the pathogen, we can track and characterize the pathogen evolution, and by doing so by identifying mutations that are linked to viral properties or to the way those pathogen interacts with their hosts. As much as we can do drug resistance studies, where the question here becomes, does the pathogen have mutations or that affect drug resistance, or do they have drug-resistant genes? They are acquiring or losing those genes. So having a fast and informative, reliable mutation detector in known gene targets in this case is the way to go. And again, NGS can provide us with that capability to, especially for discovering novel mutation that can give us an advantage over there as well. And finally, but not last but not least, identification and discovery. So what is the pathogen? What are the pathogens that are present in a certain sample, in a certain specimen? So identify multiple pathogens in a single run, maybe with the ability not to have to isolate those pathogens or to give too much care about what is the in the experimental part of what is the host genome. And again, here discover novel variants. Here it's and that's the point of all this presentation here is where the White Glove team and the White Glove service comes in. And in general, where ion AmpliSeq NGS panel can be very useful for infectious disease. This is a technique, is the gold standard technique for targeted sequencing and NGS library preparation, and is based on a very well-established design pipeline to create those panels for those libraries. We can roughly, let's say, synthesize the types here in ready-to-order, what we call it semi-custom and fully custom solutions. So ready-to-order solution, and I'm staying in the realm here of infectious disease, can be purchased directly at AmpliSeq.com. You can review them for free. It's free access. And they are the AMR panel, the TB research panel for tuberculosis, the pan-bacterial research panel, and the Ebola research panel. Then we have solution that we call here semi-custom. Semi-custom lives directly into the website, in the same website, and give you the opportunity on combining different things together. Now, semi-custom is because this is more related to human applications, where for the, we have a pre-designed content, gene-based pre-designed content that can be assembled in different ways. But it can also be with certain limitation used for infectious disease research, as long as you have one reference sequence that define and represent what you want to study, what you want to sequence, which most of the time is not the case, unfortunately. So we were talking with pathogen and infectious disease, we're talking about species with different strain and different and a very high level potentially of diversity to be covered. And with this approach, we need to be able to have the information on that diversity to be able to design a panel that captures that diversity the way it has to be captured for the different goals that we have seen before. And this is where AmpliSeq White Glove service comes in. This is really more than a tool. This is more of a partnership. It's a whole team that is dedicated to designing the panels. It's the same team that develops the design strategies for these panels and maintains the design pipeline. And so this process, I'm going to give you a very high overview of how it works and how to access that. It's actually involving manual curation, more than curation, manual interaction, human interaction, and where we have to come to discuss detailed specifications, QC the input in a specific way for very specific designs and very peculiar designs. So a little bit about the when you want to access this sort of service and why we do provide this sort of service. This is a service that was created with having a high specific research question in mind. So to provide the answer to those high specific questions. So there are here summarized 5 reasons, but we can go on forever with different reasons for that. But just as an example, into the into the infectious disease realm, your pathogens might vary regionally or seasonally. It may be different from different countries, or it may be different from this year to next year. So you need a fast response, and you need to be capturing that diversity. Or you are studying something which is novel, it's emerging. There are sequences and similarities, and so on and so on and so forth. Or in general terms, you are focused on one or more targets, or your what you want to study is more pathogen at once at the same time. That's the power of NGS versus existing other techniques, which are done one by one, etc. That's when accessing this kind of service, and when providing for us this kind of service, is a sort of privilege for us to be able to address your research need. So here is where I'm introducing very, very quickly the AmpliSeq white glove service. So again, this is meant to design solution tailored for your research need. It's handled entirely by a super specialized R&D bioinformatics team. So this is part of our R&D at Thermo Scientific. They dedicate to create the custom AmpliSeq solutions through the white glove service in this case, but also they are the ones creating all AmpliSeq designs for Thermo Fisher. So all the fixed panels and other products you can find on thermofisher.com. And at the same time, as I mentioned, they are responsible for implementing, maintaining, and improving the design pipeline. So they are really knowing this stuff in and out. So this extensive knowledge is put to, and experience is put to service for many research applications. We have the ability to create, they have the ability to create custom methylation panel with targeted methylation, custom pharmacogenomics panels. But coming again to infectious disease, they can create whole genome sequencing, viral and bacterial, species detection panels, again, viral and bacterial, and panel for species typing, which are the 3 main, say, arenas where this works for NGS. How the process itself works. So the process leverages on the existence of this R&D team. And basically, the first step is up to the user, it's up to the customer. So our customer has a special research need. So the first step is to bring it up to us. So to your regional channels. It could be your sales rep, it can be your FAS, or your bioinformatics support from Thermo Fisher. You can have a conversation with that. That's step 1. After step 1, we will be contacted, and the white glove team will be contacted and say, hey, we have this customer with this special need. More often than not, we go on with it, setting up what we call a pre-design call with one or more representative from the white glove service team and the customer and sales to try and figure out more of the details, whatever hasn't been discussed in the initial discussion, and to figure out, at least the general details and the scope of the design, because those will definitely start defining what the eventual outcome will be for that. So the actual design and the actual panel in your hands. Once this discussion is done, and if you decide to go ahead, then the request is submitted internally by our sales rep. So your job at that point is done. And the request is basically reviewed for consistency and approve. All the data is there and so on. This is more a formality. It's always most of the time done within a week. And then at step 5, the design which has been approved, the design request approved, gets into what we call the design queue. This is a time where we have, of course, a certain number of people working on several designs. And so it's just the waiting time for having your specific request assigned to the most adequate partner in the white glove team to carry out the design. When that happens, the request is assigned to a white glove lead, which is step 6 here, then before starting the design, even if we have all the information, we are still putting them down point by point and ask you through sales or whomever have submitted the request to confirm that. That exercise is an extra exercise, but it's needed. So we want to have 100% certainty that what you're asking us to do is exactly what we understand you're asking us to do. And that's the finalization of those requirements for the design. Then the design begins. The design time depends on the complexity of it. It can be just, a very simple design with a very simple structured virus, for example, or a multi-virus design combined with something else that get, a longer time. It's usually in the terms of some weeks. Okay, so when I'm talking long stuff, it's not. I'm always talking about a few weeks. Once the design is completed, the only thing we ask from you is to have an active account on AmpliSeq.com, and that's where magically that final design will appear with all the files necessary to set up a run on ion torrent instrument to produce the data and the sequencing data. So that's just a very large overview. If you want and you get entered and you're interested, you have one of such design, go to your sales rep, and we will go over this, seamlessly, because this is very well established, and you will get all the details, because those details are worthless here, but are very much more sensate when we are discussing a specific design that you have. Just a couple of tips, actually two tips here, very general to speed up and make this process smooth. These come more from experience than anything else, and tip number 1 is finalize and clearly define your design requirements. So there's nothing worse than having a misunderstanding somewhere. You want something, we understand something else, or to add something at the last minute after the finalization, because most of the time you see the design exercise needs to start from the very beginning. So let's define the target very well. Let's define the scope. Let's define what the input quality is, for example, and all these details. That's a good practice to have. Tip number 2 is link your institutional email address to an active AmpliSeq.com account. By institutional, I mean nothing like the commercial third-party generalistic email domain like Gmail, Yahoo, or you name it. This is because it's much easier for us to track the institution, to keep track of the institution, and it's much easier than to share the same design within your same institution without too much ado, because we will look immediately at the email domain, and we can just do that transfer without further ado if that comes necessary. Again, this is out of experience from previous design and previous submissions. Last 2, not gonna get too long on this one, but promptly respond to confirmation email, especially to the final confirmation email. It's not necessarily directed to you, but be in strict contact with FAS and sales on the status. Be responsive of those, because if we don't lose any time there because we understood everything correctly, then the design will start immediately, and it's a great advantage. And number 4, finally, identify and work with our support if needed to provide the proper data definition and format. The design eventually will depend on the quality of the input data, and it will depend as well on the format, on the proper format. So providing a sequence in a FASTA file, it's something unambiguous, doesn't need any QC of any sort. If it's provided in any other format, then there is an extra step that needs a little bit of back and forth, just increasing the turnaround time for the whole project. But that's all. More or less, we are set with this very, very simple common sense rules here. Said that, so giving you a little bit of overview of how does this work. So try it out. It's okay. I'm just giving you 3 very quick example of things that have been achieved using panels designed by the AmpliSeq white glove through the AmpliSeq white glove service. Application 1, the example for multi-species detection. So in this case, the design strategy is to enrich genomic region of interest to detect pathogens in clinical research samples. So you have samples, you want to detect certain pathogens in those specimens. And then the idea is to prepare a library that will specifically enrich for those. And from the data, you can figure out what pathogen is in or is not in that particular sample. In this case, it was done for detection of bovine pathogens. So that's also one thing we do. You don't need to, you need to specify as needed the host species. That's very useful for the design because we can check that the primers that compose the panel will not interact and will not amplify the host species. It's what we call exclusion genomes, right? So you can go ahead directly from the sample. And in this case, it's just, the bovine reference genome. It could be human, it could be anything else. It's just to give you a general example. As you can see, this NGS method provided here, the detection of these organs, even when qPCR threshold were in the 30s, so very low copies. And then, bottom line is that the results here demonstrate that it is definitely possible to incorporate NGS in a cost-effective ways in a research laboratory for pathogen detection. It could be veterinary, or it could be some other field of your interest. Application number 2 for the second example of 3 is instead related to the molecular epidemiological studies. So in this case, the goal is to determine the phylogenetic relationship of a certain virus, for example, or pathogen in general, circulating in a certain area. In this case, is the relationship with canine distemper virus, CDV, circulating in the US. Here you see the publication reference. Again, this was achieved through an AmpliSeq white glove service team design panel. Here, multiple primers were designed specifically to amplify various strains. So we are in a situation here where we have a species, but there is a very high variability in 2 strains in that species. And so the information were provided upfront, and then the design took care of handling that variability to give you the ability to do that detection, whatever strains come up in your system. And again, bottom line here in this paper was that the continuous surveillance is required for monitoring this particular virus in the US because it came up, as something different that was seen before, as it often happens for humans as well. And then so to prevent potential vaccine breakthrough when, the pathogen can just find an escape way through our immune defense system. Last application, and then I'm almost done here, is viral genotyping. The last example I wanted to give you. In this case, we want to examine the transmission pattern using full genome sequencing. So the design strategy here is different. We have a reference for the whole genome. We have references of all the strains that are within that particular species. And we design, or the white glove team designs, primers to cover all that genome, taking in account all the variability. So using, for example, as a simple example, common primers where whenever you have a common structure and sequence among those strains, and using peculiar and specific primers as strain-specific primers for the rest. So that when you prepare your library and you get, your sequencing data, you can achieve a very high genome coverage, as has been done for months in this case for this paper, and then with a very good detection limit. In this case, they are mentioning CT34 in qPCR, and without the need to isolate or pre-amplify your sample. So you just take the specimen when it comes from and apply the panel preparation method directly to it. This was specifically done against the human genome and the human transcriptome. So this specificity where the amplicons goes and detect and define the whole genome sequencing is very much high. And again, the bottom line here to point out is that, again, genomic surveillance by NGS is straightforward, and it's a powerful tool for epidemiological research, which is always ongoing there. Just to summarize then, when it comes to infectious disease, we have different research area. There might be more than this, but in general, epidemiological research, drug resistance research, and identification and discovery. It's kind of a 3 example I just gave you. For all of this, we have ready to use panels that are highlighted here in light purple, and also custom solution, fully custom solution that were created for these or other customers for other species. I'm naming here HCV, mumps, which was the example before, Zika, HIV, full genome, or drug resistance, CMV, and so on and so forth. So just to give you an idea, this is an approach that can be used to address these questions in your research studies. To conclude then, simply NGS has transformed our ability to identify and understand pathogens. We can do that through detection. So we detect something is there and we name it. We define what it is by the sequence information at the same time. It has many applications. So we can identify microbes within mixed populations in a very specific way. It can perform research on retrospective outbreak samples, for example. You can have information on the presence of virulence factors that emerge in a certain population or in a certain infectious disease, and also discover mutation by the information that is included in a sequencing that are associated with drug resistance or other characteristics you want to study. So all these are highly specific research questions. Now I'm generalizing, but each of these, if you think one species or another, becomes very specific. And these specific questions require a very specific custom NGS solution and a very specifically effective custom solution like the ones that AmpliSeq can provide in terms of chemistry and design, and the AmpliSeq white glove service can provide in terms of human interaction, in terms of what, again, I'd rather like to think about as a partnership between you and us to come up with something which makes, in this case, our world healthier, potentially. So we are partnering with you. So just consider this. I just wanted you to make very quickly aware of our capability in design and coming to you with solutions for these sort of things. So go and if you have one of these research projects in your hand, in your head, on your back in your hand, just go out and discuss with your FAS, with your sales representative, and let's keep in touch and have a conversation, and let's start the process, and we can provide you the panel you need for. And with that, I'm open to questions.